Hello, my name is Marissa Schroeder, and I did my summer research project on the analysis of antibiotic abilities of bacteria isolated from a multi-species biofilm. So for my project, I worked with biofilms, which have been recently understood as the normal state of bacteria in the natural environment. A lot of research being done with them focuses on plasmid transfer, as biofilms have been understood as sort of one of the main routes of plasmid maintenance within bacterial populations, as they get passed back and forth between cells or species. Plasmids are instrumental in spreading antibiotic resistance, as they can transfer genes from one species of bacteria to the next. I focused on studying the impact of biofilm formation on the production of antibiotic compounds within a multi-species biofilm. So the two bacteria species I worked with were identified as a Pseudomonas species and Deinococcus aquaticus. This project used a series of assays to assess the ability of these two species to produce antibiotic compounds both individually and synergistically when they grow in a biofilm together. Organic extractions were done to isolate any molecules these bacteria produced, and then disc assays were used to analyze the ability of these bacteria to produce compounds with antibiotic effects. So biofilms form when planktonic bacteria adhere to a surface or to each other, and they begin to divide, and they start to produce an extracellular sort of matrix component that they live within. These biofilms begin to grow, and the bacteria continue to reproduce and reach a really high concentration, all the while producing this sort of extracellular structure. Finally, when a bacteria reaches a good size, chunks can break off and disperse and form a biofilm elsewhere, or bacteria can return to their planktonic life form and travel elsewhere as well. So the reason biofilms are so important is because they're frequently the sites of the spread of antibiotic resistance genes, which obviously have very large implications for human health. But bacteria living in a multi-species biofilm, so in really close contact with each other, can pass different plasmids back and forth to each other, and they can share these genes that we prefer they don't have, but to the bacteria improve their survival. So it's important for us to study and understand what biofilms are, how they help bacteria share their genes, and more along those lines. So for my project, as I mentioned before, I worked with a multi-species biofilm containing a Deinococcus aquaticus species and a Pseudomonas species. This is an SEM photograph, so a scanning electron micrograph image of my two species when they were grown in a biofilm together. So as you can see, the round Deinococcus is in there, as well as the Bacillus Pseudomonas. So as you can see from this picture, they grow quite readily next to each other, and they start to form those sort of extracellular components as well. So these are the methods I followed for my project. It started with the isolation of bacteria from an Ontario lakewater sample. Following this, identification was done through 16S sequencing, and then also selecting bacteria that can form a biofilm together and coexist in one. Once I had my two species, I did an organics extraction for both of them, and then when they were grown in a biofilm, I did an organic extraction on that as well. And then those extractions were used on disc assays to assess the ability of those bacteria to produce compounds with antibiotic effects. Finally, those extracts were also used in an FPLC for fraction collection to separate different kinds of molecules. So for my disc assays, this is an image of one. This was done using the tester strain Bacillus subtilis. So I grew a lawn of the bacteria, put a disc down, add the extract from my species onto the disc, and then see what the zone of inhibition is, or the zone where the tester strain bacteria died. So in A, I added 20 microliters of the extract that I extracted from my two species, and then B was 30, C was 40 microliters, D was 50 microliters, and then E was a positive control of tetracycline, and F was a negative control of methanol. What's important to note from these disc assays is that the size of the zone of inhibition, measured in millimeters, is increasing with the amount of extract that was added to a disc. This is a graphical representation of that. So for the 4 tester species I used, Bacillus subtilis, E. coli, Staph aureus, and Pseudomonas aeruginosa, you can see that the zone of inhibition size increased with the volume of extract added to the disc assay. You can also see from these graphs that the tester strain that both bacteria species I worked with most effective against was the Bacillus subtilis, but they were effective against all 4 of the tester strains. It's also important to note that the combined bacteria extraction showed to be just about as effective as the 2 different strains I worked with. So when they're grown together, they still are just about as effective as when they're grown separately at producing antibiotic compounds. So the next step in my project was FPLC work, and fraction collection. So the FPLC separates the molecules in my extracts basically into 50 fractions. And while it does that, it also measures the absorbance of the sample at 4 different wavelengths. So at 215 nanometers, a high absorbance indicates the presence of organic compounds. At 255 nanometers, absorbance indicates the presence of nucleic acids. At 280 nanometers, measures protein content. And then at 495 nanometers, it measures large organic compounds and conjugated compounds. And after running a sample through an FPLC, you get a chromatogram to show you what your absorbances look like in different fractions. So as you can see from these chromatograms, to start off at the top, the darker purple seen here is the organic compound seen at 215 nanometers. You can see there are spikes early on between fractions 5 and probably around 20. So these are what we expect to see following doing an organics extraction on bacteria. But it's also interesting to see that these blue and green lines further down, they indicate the presence of nucleic acid and proteins. And there's quite a decent presence of them here, a bit of a spike around fraction 27, and then a bit of a drop off. So that's interesting to see. And then measuring at 495 nanometers, the red line here at the bottom, there wasn't much seen there, which indicates that there wasn't a high quantity of larger organic compounds present in the sample. So this was my Pseudomonas species. The next one is the Deinococcus species. And you can see sort of the same pattern. There's a very early presence of small organic compounds. As you can see in these spikes, it's not quite exactly the same, but similar to what we saw in the Pseudomonas. However, the Deinococcus species doesn't have the same quantity of nucleic acid and protein prevalence that the Pseudomonas species did. So there is still some peaks, but they're not as clear and they're not as large as we saw with the Pseudomonas species. And similar to the Pseudomonas species, though, there's nothing being measured at the 499 wavelength, which indicates there's no presence of large organic compounds. The third chromatogram at the very bottom is when both species were grown together, so the extract from when they were grown together. And what's interesting is that it is different from the previous 2. If the bacteria had no reaction whatsoever to being grown together, then we would expect this third chromatogram to just look like the previous 2, directly overlaid. But that's not what we see. We see similar peaks of small organic compounds, but the differences really come out when we look at the other wavelengths and what they're measuring. So there's more of a nucleic acid and protein quantity here than there was in the Deinococcus, although it's not as large as we saw in the Pseudomonas fraction. But it is still here. There is still a peak right around 27, fraction number 27. What's most intriguing is this peak at the 495 nanometer wavelength, which indicates that there is a quantity of large organic compounds in this fraction that wasn't in the previous two extractions. So it would be very interesting to isolate that and see what molecules are there. So from the results of my summer research, I found that the disc assays results indicate that both bacteria species produce an antibiotic effect that increases with the volume of extract used. The next step to this would be to continue doing disc assays and fraction collection, different types of chromatography, to really isolate what molecules are being affected then, purify them, and see how they compare to typical antibiotics. The next result from this summer research was those chromatograms that indicated that there are differences in the production of organic molecules when the two bacteria species are grown in a biofilm together than when they're grown individually. So next step for that would be to understand what changes are occurring in the genomes of those two bacterias when they do grow in that biofilm together, what pathways are being upregulated, downregulated, what those molecular changes are. So I would like to thank the College of Health Sciences and Technology SURF program for providing funding for this work over the summer. And I'd like to thank my mentors, Dr. Bill Thomas and Dr. Andre Hudson, for their guidance and support.